Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjon classes. Dear students, we are in a continuation of the one of the important topic of principle of inheritance and variation, that is sex determination. In previous classes, we have already talked about the genic balance theory and the chromosomal theory of sex determination. Today, we are going to talk about the environmental regulation of sex determination. Now, when we talk about environmental regulation of sex determination, it means here the environment, if we talk, have very have very significant role in have a very significant role in lower organism with respect to sex determination. Okay, so in lower organism, it is actually playing a very great role. So if we talk about environment, then the example in which you will see the environmental regulation of sex determination that include some of the examples like Vanellia, Crepidula, and Ophryotrocha. Okay, so in this, that is Vanellia and Ophryotrocha, actually it is CHA and Crepidula. In this, you will see that the environment playing a great role in determination of sex. In this example is important, which is known as Vanellia viridis. This Vanellia viridis is a marine worm, and in this, their larvae are all similar. Their larvae are all similar. Okay. So, in Bonellia viridis, which is a marine worm, Balzar was a scientist who observed this mechanism of environmental regulation in Bonellia viridis. Observed by F. Balzar in Bonellia viridis that the larvae are all similar, they are undifferentiated, but that when larvae settle down in mud, when larvae settle down in mud, it grow up and it grow up into mature female. But those larvae which settle down near proboscis of female, proboscis is a head part, they become they develop into male. Suppose this is the female, and this female, if we talk, it is large and free living. This is free living, and this is a particularly we can say male. Now this male is parasite on female, and this male is small in size. Okay, so when the larva mature near, or this is a proboscis, so when larva settle down near the proboscis or head part of a female, then it develop into the male. And if larva is far away from the particular this female, free living female, and settle down into mud, then it will develop into the what? A particularly a female. So such type of sex determination is observed by. Now why it is happening? Because it is observed that the proboscis secretes some hormone which induce a maleness in larva. Okay, so this way it is observed. Now, if we talk about another example, some more examples for this, then in which show the environmental sexual determination is seen, then it is crocodile. Crocodile, and specifically we can say lizard. In both crocodile and lizard, if you observe that there is a high temperature, if larvae settle down near the high temperature, then they develop into, they cause maleness. If there is a low temperature, then it induce or it cause femaleness. Okay. One more example is of turtle. In turtle, if temperature is less than 28 degrees Celsius, it induce maleness. If in turtle the temperature is greater than 33 degrees Celsius, induce femaleness. And if the temperature is between 28 to 33 degrees Celsius, then male and female are in equal number. Okay. One more example is of a medusa, which is a marine fish. It has a sex change ability, and in warm water it is female, and in cold water it is male. So in this sex determination, environmentally play a role in case of this, some of the examples. Okay. Now after this, some of the examples in environmental regulation of sex determination. The next one which we are going to discuss, that is hormonal regulation of sex determination. So let's talk about the another form of regulation of sex determination, that is the hormonal regulation of sex determination. When we talk about hormonal regulation of sex determination, here we understand that there are some sex hormones. Okay. There are presence of some sex hormones in organism, and this sex hormones are have a important role, have more important role in sex determination. A more important role in sex determination rather than, actually we can say, sex differentiation. A more important role in sex differentiation rather than sex determination. Now the question is why? Because they influence how it is going to develop as a male or female. They hormonally induce the differentiation that is not going to determine. Okay. So here we are going to discuss two examples. First example we are taking free martin in cattle. Dear students, if we talk about free martin in cattle, see, in cattle, when twins are produced, one is male and another is female, and if during development they are connected by common placenta, then they will be of this type. Suppose here if I talk, what will happen? So suppose this is the particular cattle, okay, and this is another cattle, and both are connected by a one common placental membrane. Okay, these both are developing. This is developing as a female, and this is developing as a male. Okay, these two twins are getting produced. Here, what happens? Male develop first, so it is normal, but when female develop, it get influenced by male hormone received through common placenta, induce some male character in female cattle. So if in female cattle it is going to induce some of the male character, then here these two opposite sex twins, in which the male is normal and the female is going to be sterile. But this sterile female with many male character, because the hormone from the male has influenced certain male characteristic in case of the females, because of this female is sterile. And this sterile female is known as primary. So this sterile female are known as free Martin. And in this free Martin or sterile female, you will see the some of the poor female characteristic, like there can be undeveloped ovaries can be there, or there can be a non-functioning ovaries can be possible. So this is the occur in case of the free Martin, in case of catas. But now another example which we are going to discuss, another thing which we are going to discuss here, that is gynedomorphism. Gynedomorphism. Gynedomorphism is also known as sex mosaic mechanism. And in gynedomorphism, gynandro actually gynedomorph are known as sex mosaic. Gynedomorph are produced. These gynedomorphs are rare and special type of organism which have half male body and half female body. Why? Because this male body have AA plus X0 type of cells, male part, and this female body has AA plus XX type of chromosomal constitution. Here 0 represent what absence of one X, and out of two X, when one get absent, this will not develop a complete female characteristic, so some of the male characteristic will be same. Means this is due to, this is actually some cells with the male XX AAXO condition and some with the female like the AAXX condition. Why? Because it is due to loss of one X chromosome during metaphase of mitosis. Okay, and due to this loss of character, if you see, you will see the particular half of the particularly female and half of the male characteristic. Okay, so half of the male and female characteristic develop due to the what themselves. So now where this gynedomorphism is seen. So if we talk about the example, it is seen or observed in case of the Drosophila and silkworm. So here you can see, if suppose initially in zygote AA plus XX cells are there, but during first mitosis, loss of one X chromosome occurs. So it will produce two type of cells that are AA plus XX and AA plus first mitosis AA plus XX and AA plus X0, that is absence. So this is nothing but going to develop as a female body. These cells will be of female type and these cells will be of male body. Okay. So this is the what a particular happens in the gynedomorphism due to the loss of an X chromosome. So here, what do you mean by gynander? So gynander is nothing but what we can say. It is a gynander maybe male or female, with patches of tissue of other sex on it, then it is known as what? A specifically, and this is seen in case of Drosophila. Okay, so this is the what particularly. So the question is where this one X chromosome get lost, or there where this one X chromosome goes. Okay, so it can get heterochromatized or it can get inactive, and due to that, the particular this X chromosome get what lost and develop as a male body. Okay, so this is about the gynetomorphism. Now let's talk about one small short topic which is also somewhat associated with all the different theories of the determination of sex. Uh, and here we are going to talk about cytological basis of sex determination. When we talk about cytological basis of sex determination, then it is also known as Lyon's hypothesis or doses compensation phenomena. Dear students, here this Lyon hypothesis or doses compensation phenomena, if we talk, then it says that there is a presence of a bare body or we can say reduced heterochromatized heterochromatic X chromosome. So if one X chromosome or any X chromosome is getting heterochromatized, then that heterochromatized X chromosome become non-functional for transcription or non-functional for expression. So the expression of this will not happen. And you observed that in female, you know that there is a XX condition, but in mammals, any one of the X chromosome, any one of the X chromosome, any one of the X chromosome get temporarily and get temporarily and randomly inactivated. And randomly inactivated Now this randomly inactivated or temporarily inactivated get known as facultative heterochromatization. So this X chromosome becomes facultative heterochromatin and means tightly coiled. So if the DNA chromosome get tightly coiled, it has no space for enzyme to interact, then it is not going to express. And this tightly coiled or inactivated X chromosome are observed as darkly stained body known as bare body. Okay, so bare body is nothing but our inactivated X chromosome or heterochromatized X chromosome or tightly coiled X chromosome, which is not able to express its genes. Now it is known that this bare body are observed in inner epithelial cells, inner epithelial cells of mouth and neutrophils of blood. Inner neutrophils they appear as drumstick. So known as drumstick chromosome. So what is bare body that you understood? Why it is inactivated, if we say, or why it is getting heterochromatized? So we can say to make a balance with the male, because in male one X chromosome is there, so it is inducing a maleness. But in female two X chromosome is there, then the female is going to be expressed. So when the one X chromosome get reduced, then the particular number of X chromosome will be equal in both the sex. So if we have to identify number of their body, then one formula is used, and that is number of X chromosome minus 1. So when there is a number of X chromosome minus 1, so how many bare bodies will be there in a particular different chromosomal condition, that we need to understand. So if it is a normal male, which having a AA plus XY condition, then here X chromosome is 1. So number of X chromosome is 1 minus 1. Okay, so it is going to be 0 bare body. But if it is normal female, then it having a AA plus XX condition. So here number of X chromosome are 2 minus 1, so it is going to be 1 bare body. Then if it is XXX condition, triplet, then in this X chromosome are 3 minus 1, so it is going to be 2 bare body. If it is XXY, then it is going to be 2x chromosome minus 1, so it is going to be 1 there. If it is A plus X0, then here X chromosome is 1. So 1 minus 1 is going to be 0 there. So such a way you can count the bare body sex chromatin by this formula. Okay. So I hope you understood this is a different mechanism of sex determination we have discussed. I tried my best to cover these topics. I hope you understood that. And here we end one of the important topic, that is sex determination. Okay, dear students, thank you.